lecture is the human virome in health and disease. So what I'll present, some introduction to the human virome, then a study of the establishment of the human virome, and then, if I still have some time, interrogating metagenomic dark matter for new viruses, looking for new kinds of viruses associated with humans. Okay, so the planetary virome is huge. There's something like 10 million viruses per mil in rich seawater, just astounding numbers. They outnumber their host by something like a factor of 10. So that would mean there's, multiplying it out by the volumes of the oceans, there's something like 10 to the 31 viral particles on Earth, just incredible amounts, numerically by far the most successful biological entities. And so over here are some pictures of marine phages from Lita Proctor. The human virome is similarly totally vast. So most people are familiar with persistent and latent infections. Like half of all adults are commonly positive for the herpes viruses, CMV, EBV, HSV, and others. Unfortunately, the more pathogenic viruses are often, unfortunately, common in the human population, HIV and others, 0.8% positivity roughly. Some viruses are transient, like SARS-CoV-2. Cold viruses, very common. Sometimes we deliberately infect ourselves with viruses like the pox virus, the vaccinia virus vaccine strain to prevent smallpox infection. But that's really just the tip of the iceberg. The human genome itself is some 8% recognizable fragments of endogenous retroviruses that infected the germline in the primate lineage leading to humans. And so here's a little bit of human chromosome 11. I've asked it to show the browser to show these repeated sequences. LTR is long terminal repeat, their jargon for the endogenous retroviruses. And you can see ticks all through this line, just really common, again, comprising 8% of human DNA. Still, that's just the tip of the iceberg. Our microbiome is host to numerous viruses, bacteriophage of bacteria and archaea in human stool samples, as I'll be showing you. If you purify virus-like particles and stain under a microscope, you see something like 10 to the 9th virus-like particles per gram of human stool, just incredible amounts. So in these kinds of studies, to a virome study, you might take a specimen from humans, maybe stool or tissue or saliva or what have you. Spin out any solid waste, filter to remove cells. If you're being really careful, you might do a density gradient, chloroform extraction if you want only membrane not enclosed viruses, then nucleases to get rid of what's in the mix, then break open particles and purify nucleic acids, DNA and RNA, and then sequence, and that gives you a sample of a viral population based on sequence information. And so here's a favorable prep from gut. After fluorescent staining, you can see it's like the stars in the sky, numerous particles produced. So we're starting to get a picture of what kinds of viruses reside where in the human body. It's very early days, but some points are probably going to stay true. The gut and oral viral populations are the most numerous. Here you can see some of the numbers, 10 to the 8th per mil of saliva, 10 to the 9th per gram of feces, very, very roughly. Large numbers and types of phage, commonly the most abundant, at least as far as we can tell, seem to be bacterial viruses of various kinds. You see animal cell viruses, that's the left-hand columns here, but they're typically outnumbered by phages. But still, when we do one of these sort of sequence surveys, it's still common to find that most of the reads that you generate don't align to anything in the databases. But I think this makes sense because of the huge viral populations on Earth. What it resides in, what's been sequenced and present in databases is a very small fraction of the total. Plus, they evolve really quickly. So I don't think it's too shocking that even today, most of what we sequence doesn't have a strong match in a database. We're beginning to build up a picture of factors that are affecting the human virome, much along the lines of the cellular microbiome. Diet, genetics, aging, disease, geography, cohabitation, medication, breastfeeding, all can affect viral populations, in part through affecting the microbial host, but also by other means as well. And I'll touch on some of these as we go along. The virome can influence human health in numerous ways. Of course, many viruses directly cause infections that are pathogenic. However, some viruses can trigger a protective immune response that can protect you from infection by some other pathogen. Viruses can activate signaling via well-known toll-like receptors and other molecules. This can either be favorable if it prevents another infection, or unfavorable if it's a ineffective immune response and you're affected with something else. They can modulate bacterial abundance as predators. They can transfer DNA into bacteria and influence fitness and virulence. Cholera toxin is carried on a phage, for example, so the toxicity of cholera infection depends on whether you have that phage or not. So again, we're just starting to be able to lay out some of the, broadly, the various ways the virome can affect human health. So let me now go into a thorough study we did of the establishment of the human virome. So this slide kind of illustrates the problem. So babies are born. You take their very first bowel movement, meconium, purify virus-like particles, stain, look at them under a microscope. You see almost nothing. But a month later, it's like the stars in the sky, really rich viral populations, and so on through life. So the question we want to ask is, where is all this virus coming from? And so we did a first study of babies studied longitudinally, and I'll tell you about that. First, let me tell you about the development of the virome in healthy newborns, just a quick overview of literature from us and others. Ephraim Lim didn't detect any virome in amniotic fluid in healthy pregnancies. And so what I'm going to tell you about is health. Of course, there can be viral infections of neonates that severely impair health. The question we'll be addressing is the virome in healthy infants here. Maya Breitbart did a study of one infant, no particles in meconium, but quickly found that they were measurable. Other studies have seen within 37 hours, within 2 days, they can start to see high levels of viral particles in baby stool. So the conclusion seems to be fewer no particles at birth, rapid colonization later. And then later in life, the virome remains stable. Several studies have established that. So microbial colonization of newborns, this has been highly controversial. Early studies, it was difficult or impossible to culture bacteria from placenta or meconium or amniotic fluid. So this supported the idea that the womb was sterile, the sterile womb hypothesis. Later work, PCR and deep sequencing suggested possible presence of bacterial DNA in several compartments, including the placenta, the fetus. This became very controversial. My colleagues here at Penn asked us to help them with a sampling study. And so we checked, and we could not find a placenta microbiome over the contamination background. And multiple others, including a very thorough study from the Sanger Center, have reached that same conclusion. Also, coming from another direction, you can make germ-free animals. If you explant neonates sterilely from a mother by sterile cesarean section, put them somewhere sterile, they stay sterile. You can make germ-free mice. You can even make germ-free humans, so which has been done on very rare occasions. So I tend to think that the womb is sterile and infants are colonized shortly after rupture of membranes and delivery, and that'll come up in the colonization study I'm going to be describing. So that's shown here. Again, we're seeing particles appear within 1 month and stable after that. Here's virus-like particles per gram of feces. It's low or absent early and comes on late, quantified over the 20 babies I mentioned that we studied in our first cohort. Similarly, 16S rRNA, we see little or no in most of the babies. And then colonization follows in most of the babies, then quite measurably. So I will take this and lots of other data from others to indicate that the womb is commonly sterile at birth and babies are rapidly colonized. So what is the origin of the early life virome? So we did a whole bunch of experiments. We purified virus-like particles. We counted them. As I mentioned, we sequenced them. We purified total stool DNA from the same sample series, shotgun analysis, targeted qPCR follow-up. We isolated bacterial strains from early life samples. We sequenced the genomes. We tested phage production and replication, and we did some other stuff as well. And so I'll be summarizing the results of those studies. So first, from the sequence data, we commonly saw bacteriophages as the most abundant form. We could see animal cells rarely. So each column here is a sample from a baby. Each row is a kind of virus. Over here, the names of the viruses. These are phage. These are animal cell viruses. We saw a little more animal cell virus by month 4, and I'll come back to that at the end. So we saw a lot of phage. So how are these phage growing? As you probably well know, phage can have 2 major life cycles. They can infect a bacterial cell, replicate, make new heads, tails, genome copies, explode the cells, and make new particles. However, many phage can be temperate as well. They can introduce their DNA and integrate into a host cell genome. After that, the viral DNA is copied by DNA replication, just like any cellular gene. However, if the cell experiences an adverse signal like DNA damage, you can induce, resume lytic growth, and produce a burst. So one of the questions we had was, well, how are viruses growing? If we're seeing a lot of phage, is it lytic or lysogenic growth? And what does that tell us about how they got there to begin with? Well, we isolated 24 bacterial strains from a stool of the 20 infants. We tried to plate the virus-like particles on each strain, looking for plaques. Here's our positive control. And we didn't see any. We could not find any examples of these infant virus-like particles growing when they're put back on the cells from that infant. So, by the way, something to remember is that if a phage tries to infect a cell that's a lysogen, and that lysogen's making repressors, it can't grow. So that's starting to point us towards thinking about lysogeny in this case. So a computational classification of the phage replication styles using this tool FACS suggested they tended to be more temperate versus more lytic. So again, this is starting to point towards the temperate life cycle, induced prophages rather than lytic growth. So we took our 24 strains from the babies and asked, are they producing phage? So each row here is one of the 24 strains. Each column shows the conditions we used for induction with an inducer, mitomycin C, a DNA damaging agent, or not, and aerobic or anaerobic conditions. And the number of particles produced is shown here. And you can see that over half of them, at least in the presence of DNA damage, produced phage. In fact, 80% produced substantial levels of virus-like particles. So now we're starting to see a lot of virus produced, apparently as induction in the presence of DNA damage. So we sequenced the genomes of the bacteria, sequenced the phage, and compared, and we could find in many cases the genomes of the phage integrated into the bacteria. So here's an E. coli strain from one of the babies, and here's a phage. These are lots of nice phage genes in this genetic map. And we could go back and see in the strain, either with inducer or not, we could see this phage. And when we went back to the baby poop virus-like particles, we could see this phage also. So this is really saying that this phage seems to be coming as the way it's produced. Strain comes into baby, it harbors a lysogen, that phage induces, it has a burst, and that produces the virus-like particles we saw in this case. Okay, but how do we know this is really accounting for the full population? Well, strictly speaking, we don't know that it accounts for all of it, but we could see that as bacteria were more abundant in the baby's poop, so the virus-like particles encoded by that bacteria were more abundant in that baby's virus-like particle prep from the stool sample. And we could get up to explaining like half the viruses in favorable cases. So that really makes us think that this is the main mechanism that we're seeing. So we think prophage induction from pioneer bacteria provides an explanation for the source of the observed early life virome community. Later, we could see more lytic phages, but early on, we think bacteria colonize baby. They commonly have integrated prophage. Those induce periodically, and that's the first population of viruses that we're seeing. Later, I mentioned we saw more animal cell viruses, and as we began to review our metadata, we realized there was a strong association with breastfeeding, where breastfeeding led to less recovery of virus-like particles. So here, yellow shows breastfed, green shows formula only, and here are a bunch of different kinds of viruses, adeno, anello, astro, da-da-da-da. And you can see in the first 20 babies, we see a strong greater abundance of viruses in the formula-fed babies and not the breastfed babies. This didn't achieve statistical significance because of the small numbers. However, we did another cohort from the US with 125 new babies and saw the effect again, and this time it's statistically significant. And then we went out and got some African babies and studied them. In this case, we used qPCR. We validated with our 2 US cohorts with qPCR and got the same answer. And then for the African samples, we had only rectal swabs. And so we attempted qPCR only for those samples and got a strong effect of breastfeeding being suppressive of virus accumulation, formula-fed babies having significantly more virus-like particles. We could also see an effect on phage populations. It's well known that breastfeeding can lead to outgrowth in babies of several bacterial lineages, Bifidobacterium, Lactobacillus, and their associated phages were also enriched in abundance in those babies, and so an effect on phages as well. So just to summarize this part of the talk, viral colonization in early life we think is stepwise. Healthy babies are usually sterile at birth. The first phase is characterized by induction of prophage from pioneer bacteria. A subsequent step involves colonization with viruses infecting human cells, and this is modulated by breastfeeding. And the protective effects of breastfeeding could be validated in cohorts from both the US and Africa. And one nice thing was we saw a significant effect of mixed feeding of breast milk and formula. It was suppressive of animal cell viruses, and so that's sort of nice encouragement for new mothers to breastfeed as much as possible, even if you can't exclusively breastfeed. Okay, in the last couple minutes, let me tell you about one other study interrogating the metagenomic dark matter for new viruses. So some viral infections are highly pathogenic. Other viruses are very common in humans, but have no known adverse effects and thus appear to be benign commensals. And so this includes anelloviruses, small circular single-strand DNA viruses, and I'll be telling you about a group we found called Redondoviruses. These would be unknown if not for the metagenomic methods. As I mentioned, when we sequence, commonly a majority of the reads don't align to anything obvious. So what the heck are they? We think they're commonly viruses, often phage, that haven't been studied closely before. But here I'm going to tell you about a new family of viruses that we found that we think are kind of interesting. So we started studying human airway samples during lung transplant. And so we had 2 donor-recipient pairs. And the students who are doing this work noticed a little bit of a hit on a circovirus. And these guys are actually significant as agricultural pathogens. They're pathogenic in pigs, members of the Cercoviridae lineage. So that caught our attention. And so aligning to databases, it turned out that the reads they found aligned to a little bit of this little studied pig virus, porcine stool associated circular virus 5. It seems to be a crest virus, a circular rep-encoding single-strand DNA virus, pretty much unstudied. But they found that when they tried to assemble reads, they got some very nice genomes. So that's shown over here. Here's a Redondovirus genome. So first they assembled 2 genomes from these lung donors. Then using those as probes against our samples from other respiratory samples, we found 17 genomes. And also another group reported 2 of these, Coy et al. In 2017. They seem to be members of the CRESS viral group. They encode a capsid and a rep protein, replication initiation protein, and also a third protein of unknown function, or 3. The students named them Redondoviridae, for Spanish for round. They're 2 species. They seem to be strictly human associated, respiratory and oral. So they did a very thorough search and found no representation of these viruses among CRISPR spacers. So they don't look prokaryotic. Also, they don't have prokaryotic Shine-Dalgarno sequences. So we're thinking they grow either on human cells or a eukaryotic organism associated with humans. So they then followed up with analyzing over 7,000 metagenomic samples, and you can say a lot after that. So Redondoviruses, it turned out, were associated with periodontitis and critical illness. So here, periodontitis samples, you can see Redondovirus reads, and they go down with treatment in 2 different studies. And we did our own qPCR studies of critically ill patients in the hospital system here at Penn. And you could see we got some high-level colonization. This is cycle of threshold, so low means more, in critically ill oropharynx or lung, and long-term persistence in these critically ill patients. So something to do with disease. Lewis Taylor and colleagues followed up with global studies, samples from Africa, saliva samples, and elsewhere. They found positivity as high as 80%. So in some populations, so really high levels of these Redondoviruses in saliva, particularly after we got better at scanning for these things. Found globally, and so we think that, and both species seem to be found globally. So the inference is that Redondoviruses have been associated with humans for a very long evolutionary period, given that both species are very widespread. We've done studies of respiratory samples from COVID patients, and we find Redondoviruses are prominent features there also. We published a paper recently on 83 hospitalized patients, 507 samples, a pretty thorough study. Among other things, we checked, did targeted qPCR for Nelloviruses and Redondoviruses, and we found that Redondoviruses were actually significantly higher among intubated patients, and Anelloviridae were also high, these commensal viruses. And then when we wanted to predict severity of disease, either as intubation or the WHO score in COVID, the small circular viruses were actually our strongest predictor for intubation and a notable predictor for WHO score. We got pretty good area under the curves in each case. So there seems to be some connection between these guys and disease. It may, for example, it may be that when in an inflammatory condition, it's favorable for outgrowth of these viruses. So we're presently trying to figure out what cells these grow on. It could either be a human cell or some human-associated eukaryote, which actually is the model we're favoring right now, and trying to get final data on this. So stay tuned. So, summaries of forays into understanding the virome dark matter. Often much of the sequence in a virome study doesn't look like anything in a database. Not surprising, given the vast numbers of global viruses and the speed of evolution. Some viruses are quite common in humans, but benign, and wouldn't even be known if it weren't for the metagenomic methods, the Anelloviridae, Redondoviruses, and others. So one example, our study of Redondoviruses started with a weak hit of lung transplant samples, and it's led to a bunch of interesting follow-up. They're globally ubiquitous, enriched in subjects with periodontitis, intubation, severe COVID, and we're presently hunting for the host cell, trying to figure out where they're growing and how they're doing it. So that concludes what I wanted to say, an introduction to the vast human virome. It's so cool, I think, that in every study today still, most of what we see commonly is stuff we've never seen before. Just the amount of novelty is really cool. The establishment of the human virome, pioneer bacteria commonly have integrated prophages. They induce, that's the first wave. Then we can see some animal cell viruses, but they're suppressed by breastfeeding. And then metagenomic dark matter, hunting for new viruses, really, really interesting and fertile area for new discovery, I think.